Howdy folks, thanks for checking into MrUlrich'sNanobiology.com. I am Mr. Ulrich. Past few classes we've been talking about cell parts. In fact, last class we talked about the cell membrane in particular. In this little video notes casty thing, I'm going to be going through how material gets into and out of cells. We should probably talk a little bit about diffusion, which is what's going on in that little box. The dots there starting out in the lower left-hand corner. We're all nice and concentrated down here, and out here there was no concentration. And over time, those molecules were jumbling about, bumping into one another, and eventually ended up spreading out. This is an example of the second law of thermodynamics at work. That's the thing that says that the universe tends towards a higher state of entropy. Entropy is just a froofy way of saying disorder. And I guess this is another way of saying that chaos reigns supreme, or that all things fall apart. But when we're talking about transport of material, we're really talking about material moving from a region of high concentration, back when they were, all those little dots were down here, to regions of low concentration out here in the outside. Simple diffusion then becomes the most basic way that material gets in and out of cells, not just how it moves around within systems. In this diagram, we have molecules of dye on this side and no molecules of dye on the other side. This dotted line represents a semi-permeable membrane. And of course, these molecules of dye here are moving. Right, so they're bumping into one another, ping, 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 in a great huge billiards game. You can think of it like that. Boom, 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 boom. All these things are bumping into one another. And as they bump into one another, sometimes they hit the membrane and they bounce back, but other times they'll fit through the wall of the membrane. And that means that over time, we're going to end up with some of these molecules ending up over here as they bump into one another. All right, and they're starting to spread out, just like the purple dots in the last frame were spreading out. Now they're spreading out with a little membrane in the way. And this might slow it down a little bit, depending on how big the holes are and how many there are. But really, it's not going to keep diffusion from happening. So diffusion is going to happen. Eventually, as the things move from high concentration to low concentration, we're going to end up with equal amounts on one side as we have on the other. It does not mean that the molecules stop moving. Of course, the molecules are still moving. It's just we have equal amounts moving in from one side as we do from the other. So this is a good example of static equilibrium. Because the cell does no work to move the material, it's really the kinetic energy of those molecules themselves that's moving them across. We call this passive transport because, like I said, the cell expends no energy to get the material to move through that membrane. They're just bouncing into one another. Simple diffusion is all well and good if you're the type of molecule that can actually make its way through the cell membrane in between the lipid molecules. If you remember, this area here, it's made up of all of the hydrocarbon chains of the fatty acids, is hydrophobic, and that means that any polar molecules out here, if this is a polar molecule, is not going to be able to make it through this layer. That layer is basically impermeable to polar molecules. So that's where facilitated diffusion comes in. Facilitated just means with help. And of course, the molecules that are doing the help, the molecules that do most of the work, are those proteins. In this case, it's the integral proteins that are providing channels by which specific molecules can make their way through the cell membrane. They have to be the right shape. They're not just open willy-nilly so that anybody can get through. Important thing for facilitated diffusion is that the cell is still not expending any ATP. It's not burning any energy in order to get the material to go through. The materials are still moving from that high concentration to the low concentration. So the movement really relies on the kinetic energy of the individual particles bouncing into one another and finding their way through the holes. Still, these remained open. They're not gated, so that does mean that molecules can move through these pretty quickly. But these molecules still do have some, like I said, they do only let some specific molecules through, and so in some ways we can think of these facilitated diffusion protein channels as bouncers. Facilitated diffusion, simple diffusion, these forms of passive transport work great for moving material from a region of high concentration to a region of low concentration, or with the concentration gradient, down the concentration gradient. We talk about the concentration gradient, we're just referring to the difference in concentration from one region to another. The issue, of course, is that cells need to move materials against the concentration gradient as well. In this case, the open protein channels for passive transport just will not do. We're going to need different types of proteins to do this. These proteins have a particular shape here, and they look like they're not open all the time. They have a spot for molecules to bind. And then once the molecule is bound here, a shape change can take place, and the molecule that used to be on the outside is now on the inside. So these proteins behave as pumps, moving materials, like we said, against the concentration gradient from a region of low concentration to a region of high concentration. This costs ATP. It takes ATP in order for that conformational change to take place and for the movement of, in order for that molecule to be freed on the other side. In this way, these transport proteins act kind of as doormen instead of bouncers. So, quick review here then. Passive transport includes simple diffusion, which allows nonpolar hydrophobic molecules to make it through that, the tail part of the lipid bilayer. So lipids are good for these things. It's still diffusion, so it's going from a high concentration to a low concentration. In facilitated diffusion, this is to allow polar hydrophilic molecules to get through the cell membrane through those protein channels. But it's still diffusion, so we're going from a high concentration to a low concentration. In active transport, we move things against the concentration gradient from low to high. These use those protein pumps and do require ATP. To wrap this up in picture form, up top we got simple diffusion moving nonpolar molecules directly through the cell membrane itself between the phospholipids. We got facilitated diffusion moving polar molecules through the transport tubes that are open. And then we have active transport, which is moving all kinds of different materials, but just against the concentration gradient with the utilization of ATP. So far, we've really only been talking about smallish molecules. What about the big, huge honking molecules that are out there? I mean, sometimes cells need to move proteins that are the same size, if not bigger, than the transport tubes that go through the cell membrane in the first place. So how does the cell get these things in and out of the cell? Well, it's going to utilize vesicles and vacuoles, which are basically little bags of cell membrane itself inside of the cytoplasm. If you remember, the plasma membrane, cell membrane, those molecules are not bonded to one another, so there's some fluidity, and just like bubbles, we can take a small bubble and have it fuse and join with a larger bubble, or take a larger bubble and break it up into smaller bubbles. And that's exactly how the vesicles and vacuoles work. When we form vesicles to move things into the cell, it's a process known as endocytosis. There are a couple different versions of this. Phagocytosis is when the cell surrounds and eats large chunky bits, as I described them. And then there's penocytosis, which is more like cells drinking. This is when the cell takes in small dissolved particles into a vesicle. Cells also use this to get large molecules out of the cell. We talked about exocytosis a little bit, and we talked about the action of the Golgi apparatus and the way that it makes the secretory vesicles, which are pictured down here below, in order to get material out of the cell to secrete mucus and other compounds outside. So let's take a look at the different forms of endocytosis. Up here at the top, I have phagocytosis, cell eating. And here's my big chunky bit here. And now the cell is sending out streaming out little fingers, it looks like, of cytoplasm. These are going to come around and eventually meet together. And remember, the cell membrane with its fluidity, when they meet, it'll actually join together, and this will become a kind of continuous cell membrane out here, and then this little bit will be surrounded by cell membrane, and it'll form this little bubble called a phagosome. And this phagosome is then going to fuse with the lysosome. The lysosome, of course, remember lyso means breakdown, to break, and the lysosome is filled with digestive enzymes, which then mix with the chunky bits in here and break them down so that they can possibly then diffuse or be actively transported across this across the membrane here that makes up the phagosome. Down below I have penocytosis. Penocytosis is a little different. It's kind of more generalized in that it's really little invaginations, little pockets form in the cell membrane, and material from the outer environment are kind of captured. This is a little bit more non-specific, a little more random, a little more willy-nilly. But there is a form that is very, very specific called receptor-mediated endocytosis. And in this case, it's these little, the little red Ys, the little receptor molecules. These are representing receptor molecules that are on the surface of the cell. That when the right molecule docks, it actually causes a change in the cytoskeleton underneath it, which starts off the process of invagination and making this little pocket here. And eventually the molecules that were bound will be in another vesicle. Water, the stuff of life. Water is so blasted important that we have a whole nother set of terminology that we use to talk about diffusion of water, moving from one region to another, from a high concentration to a low concentration. It's called osmosis. All right, it's just water. So that whole thing about sleeping on your textbook and learning through osmosis, total baloney, because information transfer is not osmosis. So we're still talking about moving water from a high concentration to a low concentration of water. All right, we have to focus on the water now. It doesn't matter what the other molecules are specifically. It just matters that we're talking about movement of water across a semi-permeable membrane. The key to determining the direction that material will diffuse in has everything to do with determining the concentration gradient. That is, where is there a higher concentration of the material and where is there a lower concentration? And we know the material is always going to flow from the high concentration to the low concentration. The problem is the terms that we use to describe or to identify the high concentration of water and the low concentration of water don't actually talk about the water. It's kind of reverse. We talk about what's dissolved in the water rather than how much water there is. This is kind of like when you go to the diner and you order a soda with no ice. Right? Most people will order that because they want more soda. It's the ice melts and it takes up the space that there could be more soda. And I will often ask students in class when I bring this up, why don't we just ask for more soda? Because that's really what we want. So we're asking for no ice, less ice, so that we get more soda. And the terms that we use to identify where there is more water in an osmosis type of a problem, we actually use how much stuff there is dissolved in the water to indicate that there is a lower concentration of water. Okay, so it works like this. You can have hypertonic solutions. Hyper for more, tonus is for pressure, and we'll get into osmotic pressure later. But for right now, we need to recognize that hypertonic, more dissolved stuff, means less water. So a hypertonic, you think of it like a hyper-iced soda, has less soda. So a hypertonic solution has less water. The opposite of this, of course, is a hypotonic solution. This is like when people ask for no ice. All right, you have less solute, less stuff taking up the space that water could take up. All right, and so you have more water. There's also the third state, which is an isotonic solution. Iso is for the same. So these are when there's an equal amount, not of solute and water, but there's an equal amount of solute between the two solutions that we're comparing. Also important to remember that water always goes, when we're talking about water, it's going from the high concentration of water to the low concentration of water. So it's going from the hypotonic solution to the hypertonic. The cytoplasm of a cell is a solution. It's got all kinds of stuff dissolved in it. The external environment of the cell is also a solution. Sometimes I have more stuff dissolved in it than other times, all right? But we still have a concentration gradient set up between inside of the cell and outside of the cell across that cell membrane. So cells need to be able to deal with differences in water concentration inside or outside of the cell if they're going to survive. All right, now in hypotonic solutions, all right, when we put a cell in a hypotonic solution, the cell's cytoplasm then is hypertonic when compared to the outside environment. And we know that osmosis always takes place from the hypotonic side to the hypertonic side. And so in this case, the animal cell, we put it in a hypotonic solution like fresh water or distilled water. All right, that means that the cell is going to take on water. It's going to get bigger and bigger and bigger and bigger until it pops or lyses. Right? Now, a plant cell, on the other hand, plant cells have a rigid cell wall. So when the water diffuses into the cell and causes the volume of the cell to increase, some of it's actually going to go into the central storage vacuole here, and causes the cell volume to increase, it pushes on the outside of the cell wall and creates a pressure. The pressure is called turgor. And that's actually the normal state for plants. They like to be under a little bit of pressure because that helps them plump up, and each individual cell kind of plumping up. If you think about the difference between a water balloon and a sandwich bag filled with water, you get kind of the idea. In the water balloon, the water is under pressure, and it's that turgor pressure that helps hold the plant up. Now, the opposite of the hypotonic solution would be putting a cell in a hypertonic solution, like putting an animal cell in ocean water. All right, now there's a higher concentration of dissolved material outside of the cell than inside of the cell. All right, so the inside of the cell is actually the high concentration of water. I know it sounds weird because we know there's not a whole lot of water inside of the cell, and you put a cell in an ocean, and good Lord, there's way more water outside. We're not talking about amount. What we're talking about is concentration. So there's a higher concentration of water inside of the cell than outside of the cell. And that means that the water is going to diffuse out of the cell. And in this case, the animal cell is going to shrivel on up. All right. When we do this to a plant cell, the difference is, well, the cell wall is rigid and it holds its shape. The cell cytoplasm volume gets lower and lower, and that's actually going to pull the cell membrane away from the cell wall. There's little points of adhesions in some cells, but you can see the cell membrane is, or now we can actually see the cell membrane, but the cell cytoplasm contents is getting less and less volume. Now, in an isotonic solution, all right, remember, we're not necessarily talking about equal volumes, or equal concentrations of solute and solvent, or water and what's dissolved in it. What we're talking about is equal concentrations of water inside as there are outside. And in this case, we're going to have that dynamic equilibrium reached where the same amount of water is diffusing in as is diffusing out. And that's going to take place in both of these cells. The difference is that animal cells tend to like isotonic solutions. Plant cells get kind of flabby, right? This is what causes wilting to take place in plant cells. All right, so this would be a fresh water over here in the hypotonic solution. All right, a balanced isotonic solution, kind of like what we would probably find in an IV bag, and salt water. So how do cells manage themselves in hypotonic solutions? Of course, the problem for an animal cell is that they're constantly going to take on water because there's a high concentration of water around the cell. Like we said, this is a freshwater, we can think of it as a freshwater situation. All right, and of course here the cells are going to gain water, they swell up and they can pop, and that causes big problems. All right, what paramecia do, because they live, they're a freshwater single-celled protist. All right, what they do is they have what's called a contractile vacuole. This is a specialized vacuole that takes up the water, and as it gets bigger and bigger and bigger, once it gets to a kind of fullness, there are little connections between the contractile vacuole and the cell membrane, and when that contractile vacuole contracts, gets smaller, it actually pumps the water out of the cell. This of course takes ATP, so this would be a good example of active transport, moving material out of the cell, with the cell expending energy. Plant cells, on the other hand, well, they like this, right? They want this. So when they are full, when they're in a hypotonic solution, it actually gives them some structure. Like I said, it holds them up against gravity. So here's a shot of the contractile vacuole in a paramecium. All right, there's the contractile vacuole when it's full, and there's a contractile vacuole once ATP has applied. Now in hypertonic solutions, we have a different problem. All right, now when we put the cell in salt water, the low concentration of water around the cell is going to cause the cell to lose water. All right, and this is the case with shellfish and all kinds of ocean-going fish. They drink constantly. They're constantly taking in seawater, and many sea-going marine organisms will excrete an incredibly salty urine in order to help get rid of their osmotic imbalance. All right. In the case of plant cells, they wilt. They fall over. The nice thing is all you got to do is add more water to them, and they can pop back up. So I would say if you ever have the option of choosing where you're going to drown, whether you're going to drown in the North Sea or in the Wallkill River, you definitely want to choose the North Sea. All right, because when you drown in the Wallkill River, what happens to your cells? They take up water and they pop, and that's a problem. But when you drown in the North Sea, your cells shrivel up. They go through plasmolysis. They plasmolize. They shrivel up. All right, and well, like plasmolysis anyway, they shrivel up, and there at least we stand the chance of resuscitating you and deplasmolizing in a way. Plasmolysis is what happens to plant cells, but we can reinflate those cells, and you stand a chance of being brought back. Plus, the North Sea is cold water, so that's going to help as well. Now, isotonic solutions, there really isn't much that the cells have to do. All right, this is really what we're trying to set up inside of our bodies. So our body cells are in a solution that has the same concentration of water and of dissolved material as the cytoplasm does, and so this causes no problem, and not much has to be done here. All right, so like I said, when we have blood cells, the blood plasma, the fluid that surrounds the blood, has the same tonicity, has the same amount of dissolved materials as the blood cell cytoplasm does. All right, so let's put this into play in a question type of situation. Most intro to osmosis questions center around the model cell in an environment. This is part of a lab that we are going to do where we make a model cell by taking dialysis tubing, in our case, and filling it with sugar solution. This is to model the cytoplasm of the cell. And then you take that model cell and you put it in a beaker filled with another solution. And of course, the question is going to ask which way is the water going to go, what's going to happen to the cell, yada, yada, yada. Don't get confused by the complex solution here of 0.03 molar sucrose and 0.02 molar glucose. All you got to do, it's simple addition, just add the two together. So that's a 0.05 molar sugar solution on the inside and a 0.03 molar sugar solution on the outside. So now we just got to set up our concentration gradient. So the cell, is it hypertonic compared to the outside, or is it hypotonic? Hit pause while you're trying to figure out. Right. Of course, it's hypertonic because it has more stuff dissolved in it. Right. I'm hoping that that doesn't give you a problem figuring out what the beaker is, because if the cell is hypertonic, then by definition the beaker is going to be hypotonic. The trick here is, which way does the water flow? Is it going to flow in or out of the cell? Water always diffuses from the hypotonic solution to the hypertonic solution. Since the inside of the cell is hypertonic, it's going to flow in. And in this case, the bag is actually going to grow and get larger. All right, we'll stop there for now. If you have any questions, feel free to send me an email. Go to MrUlrichsNanobiology.com. My email is right on the homepage there. If you have any questions, comments, concerns, anything, send me a note. Other than that, I'll see you in class.